First of all, I would like to thank you, Dr. Wurzer, and the Green Carbon Webinar Group for giving me this opportunity, and also, the service that you are doing for this carbon group is immense. I rarely miss the presentations that are being conducted here or presented. This presentation includes information that is contributed by USDA colleagues in my lab, that is my technicians, as well as the visiting scientist, then Dr. Fizal Ahmad from Malaysian Cocoa Board. Even though that fellowship, Norman Borlaug Fellowship, was only for 12 weeks, we have accomplished a lot and the work is still being continued. So I am going to give an overview of what we have done, and with a focus on preparation of or synthesis of the biochar from cocoa pod waste, and also some characterization of this biochar and some of the work that is done to meet the objectives of the Borlaug Fellowship, that is to develop a slow release fertilizer using the biochar from cocoa biochar. And I would like to say that this disclaimer, as a federal employee, I would like to say this presentation is approved for public release. And also we are not promoting any commercial products or any particular agency. And also I need to declare that I have no commercial interest to report. And I would start with a brief outline of our organization, where I am currently working, because it's almost 2 years since I moved from US Department of Agriculture to Environmental Protection Agency. And I will talk about the focus topic for today, that is biochar from cocoa pod waste and characterization. And of course, the biomass that we used is cocoa pod waste. And biochar synthesis is using high temperature pyrolysis under anoxic conditions. Our characterization, I'm reporting only a few of those, that is the important ones. That is the surface area, elemental analysis, and surface morphology, and X-ray diffraction, or the crystallinity of the biochar, and some spectral analysis using FTIR. And I would also mention that fertilizer infusion, because our goal is, or the project objective is to develop slow release fertilizer, that is loading of fertilizers into the biochar and encapsulation, and also followed by some preliminary data on leaching studies. That is, once we have these slow release fertilizers made out of cocoa pod biochar, we wanted to study how it is leaching, and on the flow column studies. I think this part is done at Dr. Gonzalez's lab in ARS, Indiana. We have also studied different other biochars from avocado pits, soybean hulls, pennycress, pressed cake. Pennycress is the cover crop, and cocoa pods, that is today's topic, and railroad ties, which Javier has covered. Just briefly, 1 minute overview of where I am right now. I am with the US Environmental Protection Agency. I am part of the Office of Research and Development, in which there are 3 or 4 labs. I am in the lab of, this is Center for Environmental Solutions and Emergency Response, that is known as CSER. I am with the Division of Homeland Security, Materials Management. I am with the Wide Area Infrastructure Decontamination and Infrastructure and Decontamination Branch. Our focus is emergency response research, in case if there is a inadvertent or advertent chemical spills or biological or chemical or any other oil spills, what technologies and tools that we need to use. That is our focus. That is in what we had to say, we conduct emergency response research. Thanks for giving this opportunity to introduce our branch. Why EPA is interested in biochar? I think needless to say, agro waste such as soybean hulls, avocado pits, and the list of other agro waste could be maize or all the crop residues, create a waste management problem. At the same time, if they are let for biodegrade, they release a lot of methane, which is also a potent, at least 25 times more potent compared to carbon dioxide greenhouse gas. So waste management is one of the interest for EPA. Biochar can replace petroleum-based carbon in water treatment, agriculture and soil amendments, and process operations, that is developing sustainable materials. That's also in the interest of EPA. And of course, biochar could be used in oil spill cleanup. I think these things keep happening on and off through railroad car accidents or inadvertently at different locations. So we are trying to see a cheap, effective, compatible cleanup materials. So biochar could be used, and eventually it could be incinerated or used for power generation. Biochar can simultaneously help with carbon sequestration. As I said, it reduces the net release of carbon dioxide and also methane, and helps with the carbon sequestration. Our objectives for the Borlaug Fellowship, as I previously alluded, is to develop slow-release fertilizer to provide much longer nutrient release times so that it can match with plant physiology or plant needs uptake, so that growth is unimpeded due to lack of nutrients. And we also would like to see rate of fertilizer that is leached out of the biochar, and we wanted to do the kinetic studies. And fertilizer release to match crops' metabolic requirements, as I mentioned earlier. Slow release fertilizers, because most of the urea or other nitrate fertilizers, only about 40% is taken up by the plant, but rest of it goes into waste, that is either into decomposes into ammonia and other nitrate, becomes a downstream water pollutant. So slow release fertilizers could increase the effectiveness of uptake as well as reduce the environmental pollution. Now a brief history or brief summary of the current topic, that is extraction of cocoa beans from pods. As you can see, cocoa plants are grown mostly in hot and humid conditions under shade, such as palm trees or coconut trees. That's why Malaysian government is trying to promote cocoa, as the worldwide demand for cocoa beans is increasing and the cost is increasing, and that's why our chocolates price continuously keeps on increasing. So these ripe cocoa pods are taken and beans are extracted. A pod contains about 30 to 40 of these beans. And as I would like to point out that about 67 to about 75% pod husk by weight, and beans are about 20 to 23%. So there is a lot of waste. I don't know whether you are able to see this, my pointer, but anyway, you can see a lot of waste is generated, and that becomes a nuisance, a breeding ground for mosquitoes and bad smells, and it's a pollution hazard as well as health hazard for the local people. And some of it is burnt, but a lot of it is left to rot and do that. and Malaysian board, cocoa board, wanted to make use of this pod waste and develop particularly as a soil mix for the cocoa and other seedlings, growing seedlings. I think that's one of the immediate use they are looking for, which eventually help the farmers economically as well as the seedlings will be healthier. Those here I would like to say these are the dried pod waste, and eventually this is the cocoa pod biochar in a nutshell I'm just showing. Now we'll go into the details of how we made in our lab system. This is the facility that I had in my lab at USDA Peoria. We have about, I think this is, let me say, nominal 6-inch diameter and about 40-inch quartz tube in a split furnace. And we have the end connectors, which facilitate and to control the environment that is under pyrolysis. We wanted to maintain an anoxic environment. So we use nitrogen gas and at a relatively positive pressure because we want to avoid any leakage of air or any other oxic environment. And here you can see my technician Gary Kuzniak's hands here. And this is ready, and with the loaded, each time we used to put about 300 to 400 grams of dry material, that is the cocoa pod waste, and we had a temperature profile. As previously mentioned, we used to, in this particular, within a short time, we used only 500 and 700 degrees. We wanted to avoid the torrefication temperatures of 200 to 300 degrees Celsius. But in any case, we have dwell time of about 30 minutes at a rate, and also our heating rates are about 10 degrees per Celsius per minute. And eventually at the peak temperature, that is either 500 or 700 degrees Celsius, we used about an hour of dwell time. And while cooling it, we let it cool it overnight once the temperature, heating, and pyrolysis profile is over. We cooled it overnight under nitrogen environment. And it is very important that without cooling, if you try to take out, you will get, everything gets, turns into ash. So don't open the reactor until it is cool, near room temperature. We have just showing here soybean hulls, and this is the soybean hull biochar, because soybean hulls there are mountains near the ADM facilities or other US soybean meal and soybean oil mills here in US. If you look at that, those are like mountains, tons of waste, which is either burnt or used for less things. So some of it could be converted to biochar. Of course, the yield here is about 20 to 25%, whereas for cocoa pods it is about 30 to 35%. And now I get into the characterization. I think, please, I'm not able to look at a clock around. I think, yeah, I have about 8 to 10 minutes, so I'll try to make it fast. I'm listing BET surface area. For the characterization that we have done is BET surface area and elemental analysis and surface morphology using SEM and X-ray diffraction for the crystallinity of the, to study the crystallinity of these cocoa pod biochar. Also, the fertilizer infused slow release fertilizer, and of course, FTIR spectral analysis. I think here is, I don't want you to read all those things, but I will read the important points. We have done the elemental analysis of cocoa pod, that is the starting material, and the biochar at 2 temperatures, and also cocoa pod biochar with no other treatments, that is, water washing or any other things, and urea impregnated and in ammonium nitrate, ammonium sulfate, and also coating. Here at this, within the given short time of the project, we coated with polyvinyl alcohol and coated that after infusing ammonia, urea, and just for control as just water and PVA coating. And now looking at the morphology using the scanning electron microscopes, and there are two magnifications of this is the raw cocoa pod at different magnifications. And this shows this 700 degrees Celsius rinsed with water. This is the cocoa pod biochar at two magnifications. And I'm not showing all that 500 and all other things, just only a couple of things. This is the cocoa pod with ammonia infused at different magnifications. You can see some depositions inside of the condensed particulates. We believe those are particulates, either through attrition of the surface or some of it may be ammonia nitrate. Similarly, we had, as I showed the previous table, for different things, 500, 700, ammonia, urea, and all those things, with and without encapsulation with polyvinyl alcohol. Why we use polyvinyl alcohol is it is a biodegradable and easy to encapsulate. We used only very little, 4% by weight in aqueous solution, and we have used the cross-linking with the 10% sodium hydroxide finally to complete. And I have all the details in the report. And looking at the crystallinity, I think you can see the cocoa pod biochar at 700 degrees. You can see the, this is the starting material, and cocoa pod biochar at 500 degrees. So there is a crystallinity of this one, and also there is amorphous nature in these particularly. You can see that particularly at theta, from 15 to 20, you can see amorphous phase, and also from about 37, around 40 to 47 angle 2 theta. And you can see there are some crystalline peaks, and you can see 29.3 or 29.4. These are very typical of activated carbons. And you can still see biochar, these peaks, and we don't see much of the ammonia or other urea, ammonium sulfate crystalline peaks. I think there may be some crystallinity. Much depends on if we increase the ammonia or these concentrations, then we may likely see ammonia and ammonium sulfate are the fertilizer peaks which are embedded. For each of these instruments, I have the details that are available. If anyone needs, I can supply those things. What instruments, what conditions that we have used. For example, here for this one, we FDR spectra, we obtained it with the 2400, Perkin element model 2400 series 2 analyzer. That is for the elemental analysis. And this FDR spectra is also we used Perkin element system 2000. Anyway, what I would like to bring out of this one is you can see very little difference between the biochar with fertilizer and encapsulated. I have other spectra also, which for the brevity case. What I want to mention is when we have this fertilizer and the biochar and fertilizer, biochar, and PVA encapsulation, the functional groups are not changed, and the FDR spectra look alike. From this, what we can conclude is the functional moieties, that is the hydroxyl water at this high 3200 or 35 area, and also that might include amino groups. all these things remain same. What does indicate is, of course, the abundance is different because when we add this PVA things, the density or the abundance would vary. The functional groups are still the same. The point is that there are no new reactive products that are being formed. That's what. So all the ammonia and all other things are just, surface adsorption in the pores and surface area of the, the slow release fertilizer. And we have conducted slow release fertilizer preliminary studies, that is, the leaching studies. For this, we did all these things, various formulations, and we used, initially we obtained the fertilizer loading. This is, in our preliminary experiments at pH dependent things, initially ammonia concentration and nitrate concentration. These are the loading capacities, that is 159 milligrams per gram of this particular biochar, that is 700 degrees Celsius cocoa pod biochar. And here, just a summary of those things. These are the initial material. This is, we obtained this material by reverse extracting the fertilizer compounds. But fertilizer desorption studies, batch desorption, that is just using taking 0.25 grams in 100 ml of the deionized water and do time-dependent, isothermal studies, and we analyze the conductivity, pH, and also nitrate and ammonia nutrient studies. All this analysis work was done at Javier's USDA lab in the soil research lab in Indianapolis, Indiana. And also column flow studies here, where we have loaded certain amount of about 3 grams of the biochar coated material, and then column flow studies, collecting the sample at every 30 minutes and analyzing the same samples. I think with that, do I have any more time? I think I'm at 11 o'clock, I see. My time is up, and I would like to say that our future work is we have to go and study, increase the loading capacity or increase the loaded amount of these fertilizer compounds. And also, initially, we directly taken the cocoa pod biochar without any surface activation or cleanup. So we wanted to clean acid cleanup or some other kind of activation so that we could increase the surface area of the cocoa biochar, and also optimize the loading process, and look into various encapsulation biopolymers or biodegradable or environmentally benign biopolymers such as PVA or chitosan or some other polylactic acid or anything that is easy to develop a process, that is water-soluble biopolymers would be good. So those are our future work. I think Fizal is trying to do some of the work at his lab, and also Dr. Javier Gonzalez. And I am more focused in developing and further enhancing the sorption capacity of this cocoa pod biochar. With that, I'd like to thank you all for your time and your attention.
Thank you very much, Vera, for your presentation. And we are okay-ish on time, so we will have some time for questions. So you had 2 questions from John during the presentation. The first one was, did the cocoa pod feedstock showed lower moisture content than the actual biochar? And do you have an explanation for that, or was it an experimental reason, or?
I'm sorry. Can you repeat that one?
Yeah. So the cocoa pod in your table, the feedstock, showed lower moisture content than the derived biochar. Do you have an explanation for that?
Yes, we were doing the temperature and nitrogen atmosphere. Whatever the moisture to start with initially, that is evaporated during the torrefication and also nitrogen and constant nitrogen flow conditions. The moisture is, stripped off. That is one of the reasons why we see low moisture content.
Yeah. So the question was also why the moisture was so low for the feedstock. So how dry is the feedstock when you get it? Do you pre-dry it or?
Yes, those the cocoa pods we received from Malaysia, MCB, that is through Fizal, were actually pre-dried.
Okay. That explains the moisture.
Yeah.
And do you expect that loading fertilizers into biochar pores will retard the colonization of the biochar by microbiota or by fungi? So do you think you block biochar pores further when you coat them in?
Yes, that's the intent. And also the intent of encapsulation is, if we just load it and again, once it goes into water, these fertilizers are highly soluble, so they come out. So we wanted to control the release rate of the fertilizers by, using this biodegradable polymer as coating to reduce the transport coefficient.
Okay. And the question from myself was there a reason why you went for 700 degrees? I would expect a lower temperature to probably be a bit more effective for the loading.
Probably, we wanted to do at higher temperature because we wanted to eliminate all other things and also increase the surface area. At 500 degrees, our preliminary studies shows that 500 degrees to, not preliminary studies, we studied only two temperatures here, 500 degrees Celsius and 700. The properties are not very much different, the loading capacities. I think there may be an optimum temperature, which we have not done the optimum studies for that one, but it would be around 500 or 600, between 500 to 700 would be an optimum temperature that I would see for increased uptake of these fertilizers. And
so Daryl was asking, can you repeat the surface area? What was the surface area of your 700?
Our surface area, we did not do any other cleanup or activation or increasing thing. It is very low. That is about 0.2 to 1 meter square per gram. Very low. We have every belief that if we do acid cleanup or some other cleanup activation, we can go much higher.
Just because I worked with surface area measurements quite a lot, it would be interesting because the PET is using nitrogen gas for the surface area. So you will only cover pores up to 100 nanometers or so. It would be interesting to see if you have macro pores in your char.
That's one of the things we have looked at it. We wanted to go for rather instead of nitrogen, of course, helium or xenon or some other gases, but we didn't have time to do that. That would be the in future plan when we want to do it. Certainly, there are microporous surface area that we have not opened it up.
Okay, great. So we are a bit over time. So thank you very much, Vera. And